All right, good morning, class. Today we're going to get a little bit more in depth in forensic DNA analysis, which is basically we watched a little kind of segue video the other day describing the unique and wonderful things that DNA analysis have brought not only to the area of forensic science, but also to the justice system as it has enabled people wrongfully accused to be set free. So we're going to just go ahead and get started. Okay. Hopefully by the end of this lecture, you kind of have a familiarity with how crime scene evidence is collected. And DNA, we're going to talk a little bit about how it's extracted. And basically, some of the techniques that are used in DNA fingerprinting and kind of how DNA is matched. Both from a crime scene and with from an inheritance standpoint, like with parents and children. Okay, we've gone through some of the stuff from crime scenes, but right now, specifically, the evidence collected at a crime scene that contains DNA is blood, semen, teeth, saliva, hair, urine, bone, and tissue like muscle and stuff like that. If you looked at the going through the if you looked at the backseat of the car in the Kennedy, the Kennedy photos from the Kennedy assassination, you'll see that had they actually done their job and done what was supposed to be done, and there was a lot of material they could have gotten. Now, of course, in 1963, they weren't using DNA evidence, but there was a ton of evidence they could have gotten, blood evidence and stuff like that. Even though they didn't have DNA in terms of blood, they could tell what type of blood was, blood type and stuff like that. Okay, DNA evidence is individual evidence. Okay, and it can tell basically whether one individual was there and involved with what happened in the crime scene and one was not. Not to be too specific and get too deep into technical terms, but one of the things you need to know is the polymerase chain reaction technique, which can generate multiple copies of DNA evidence. Okay, there are several factors that affect DNA left at a crime scene. And one of the things, this is why it's so important to follow the rules basically for what you're supposed to do and techniques of actually collecting, protecting, and preserving evidence. So, I mean, heat, sunlight, moisture, bacteria, mold are all factors that can basically render the evidence not usable. That's why immediately as soon as people can, they need the the CSI team, the forensic team needs to get there and start collecting evidence. Okay, because as long as it sits there, especially if it's something that happened outside, in outside environment, all these things I mentioned can have an effect on that. Okay, so it's very important. And it's very important that it be done correctly too. Okay, otherwise, it'll basically be unusual. Collecting and preserving DNA is pretty much similar to doing other things. Even if we're going to collect projectiles from a gun, you always want to have gloves, and there's a lot of times people wear hazmat suits that cover their head and everything like that. Even though it might look weird or crazy, but you don't want to leave any of your own DNA on the crime scene or have it taint stuff from the crime scene. And obviously, you avoid any physical contact. Plus, in the age of communicable transmitted diseases, if you're dealing with bodily fluids and stuff, you want to have barriers between yourself and the bodily fluid. And then, like I said, you don't want to leave any of your DNA from talking, sneezing, coughing, anything in the area, hair especially. That's why a lot of people cover their hair up, because you don't even know when you're leaving hair fragments. You want to air dry the evidence. And there's a way to, some of the evidence specifically, they can cryogenically freeze it, where they can keep the evidence for a long time. I've heard of semen samples, for example, being cryogenically frozen. And obviously everything is kept in cool and dry during the transportation and storage. Once again, if any of this is messed up, when it's time to really examine the DNA and get it to the point where you can actually use it and break it down where you can actually disseminate between one DNA sample and another. Any of this stuff is messed up, it won't work. Okay, just kind of a review. Of course, I know people that had Mr. Dupree's class in the past for biology should be up on this, but we'll just kind of review cells and DNA. Where's the DNA located? If I was live with you in the classroom, I'm sure everybody who's had my class would say the DNA is located in the chromosomes, okay? And the DNA is also contained in the nucleus, as well as there are short strands of DNA called mitochondrial DNA that are actually located in the mitochondria, okay? Extraction of DNA. Extraction of DNA, there's different ways of collecting and harvesting, cells that contain DNA. When you give a DNA sample, they take a swab and rub the inside of your cheek. And that's one of the things that's become a requirement when you join the military now, for a very good reason. You have to give your fingerprints, and you have to give a DNA sample. And the thing is that it used to just be you gave your fingerprints, like I said, not to sound gruesome, but basically when somebody was lost their life on the battlefield, they would cut the tip of their index finger off, and they would actually send that back here to Washington to test next to the fingerprints they had on file to identify somebody. Now, with DNA, they have a much better way to do it. Also, you can add soap, salt solution to break up the cell membranes. Protease is a good way, if you think about it, because that is a good way to just break down and use an enzyme to digest protein. Addition, because the reason you want to digest protein, obviously, is because the DNA contains the protein. It's like the blueprint, for like the protein sequences and stuff like that. Also, if you add ethanol, DNA is being water soluble, like I said, will show up. I be honest with you, I've never seen that done. And then the PCR, if you only have a little bit of DNA, they actually use that. You'll hear that if you watch the CSI shows like CSI New Orleans or something like that. You'll hear them talk about the PCRs. Okay, DNA contains all the genetic material. Just to refresh your memory, it contains genetic material, which is for the amino acid sequence, which becomes a protein sequence, and that actually codes for all the things that make an individual who they are. Okay, and if you remember the components when nucleotide, they have a simple sugar, a phosphate, and a nitrogenous base. And there's 4 nitrogenous bases: adenine, thymine, guanine, and cytosine. And they based on the way they pair together, that's how you come up with the sequence. Okay, that's kind of a review. We beat that horse to death when we were going over DNA last year, but hopefully you still, or whenever you took my class, but hopefully you guys still remember that. All right, and then something not to forget, each human cell contains 23 pairs of chromosomes for a total of 46, which is 3 billion DNA base pairs long. So you can't look at all those. And be honest with you, people that do there's other reasons to do DNA sequencing. People who want there's certain people that have discovered plants that have medicinal purposes in some different countries. There was a place in Africa, a country in Africa, where they observed people that were chewed on this root, and the root actually, like if you had a toothache or whatever, they saw people that would chew this root and it would make them feel better. So they actually took some of the plants back to the laboratory, broke down the protein sequence or DNA sequence, and they found that it had the same medicinal characteristics of aspirin in it, and it's being used as a painkiller now. Okay. Also, so they came up with these 13 different places to look at, so you won't have to read the long sequence. And these places are called, not to get too far down into the minutia of things that, because I'm not going to require you to know all of this, but just know that the 13 places they looked at made have markers, 13 markers that kind of shorten what you need to look at to like identify the uniqueness of a certain strands of DNA. And what they came up with is that there are things called polymorphisms, which are non-coded DNA that contain unique patterns. These unique patterns repeat, called short tandem repeats, and they repeat uniquely for certain individuals. So this is the nitty-gritty of that, how they can identify things in your DNA that are like that fingerprint. Okay, only yours. And these short tandem repeats are only are individualized. They're unique to certain individuals. Okay, electrophoresis. Like I said, I wish we could do. We actually did this with shark DNA. That was just like traveling trailer that we went out, and they were parked behind where the mobile classrooms are, the temporary classrooms out there. And we were able to actually do this, like to identify a certain type of shark DNA, and it was really cool. I think it was, but it was really cool, and it's really electrophoresis is really easy as long as you follow like do what they exactly what they tell you to do, how to do it, and are careful when you do it. It works, and they had it cool because it was like this. The strands, the bands of DNA will come up on the screen. It was really cool. Okay, and so DNA profiling and fingerprinting, two samples that have the same band pattern are from the same person. Okay, and then to identify like a parent and child, in a child's basic DNA fingerprint, it must be present in one parent. So like they must have like a band that's common for a certain thing in in one parent. So for example, this is the strand, this is the whole band, all the bands that came up for like a blood stain, and so the bands have to match. Or at least one of them, and kids have to have one matching band with the parent to be considered conclusively, okay, this is your kid and stuff like that. We'll talk more about that. Okay, and then in a nutshell, what the analysis of DNA, additional things it does, obviously, and most importantly, it can match crime scene DNA with a suspect, or conversely, it can eliminate a suspect, and that is basically what happens when you see these guys that have been in jail for 29 years or whatever, and they get freed. Is that they do that. And the crime really exists is that the fact that they went to jail to begin with, and hopefully, because it's modern times and technology is even better than what happened. Maternity or determined maternity or paternity. This is a controversial one. Maternity, half your mother's DNA is a spot-on match for your half of your DNA. So maternity is like really conclusive. I'm not gonna say nothing's 100%, but like in the high 90s. Conclusively that, okay, that's your mother. The fathers are a little bit different. They can only get it to like 80%, which is good enough to say, yeah, that's your dad, but it's not like 97 or 98% for some reason. Okay, so like when there's things on TV where they are reading off whether somebody is or is not the father. It only really gets to like in the 80s for some reason. And like I said, you're definitely free falsely imprisoned individuals. And one of the things is because DNA is like exact and absolute. Okay, whereas a lot of the individuals went to jail on circumstance because they had a prior record and because a group of people said, yeah, I saw him do that, when DNA can say that the guy wasn't there, that his DNA is not his DNA and stuff like that. And then also identifying human remains. Like I said, a lot of times they use the mom's DNA to identify human remains. And if a person is missing in action, like in the military, in a war, they will get, because they have a DNA sample, they'd be able to mount, like the person's mother that give a DNA sample. If they want it, they say, we think this is your son or whatever. And then they could do it that way. Okay, we're not really going to get into DNA probes, but this is just another technique. They identify the person's DNA. They're made up of different of DNA bases complementary to the DNA strand. And if they do that, obviously if they come up with DNA bases complementary to the DNA strand, then the DNA bases are going to pair down with them. And in most criminal cases, 6 to 8 probes are used. Okay, so like I said, this is something that's used quite a bit, and that's why it's so important. Like, you know, from your biology class, to kind of understand base pairing and all that, because that's what we use. And this being said, there was one episode of CSI, Special Victims Unit, where the lead character obviously hadn't been on a crime scene, and like they said somebody made artificial DNA. Okay, technically, in the most remotest possibility, that is technically possible, because basically nucleotides, it's all chemicals, and you can link the chemicals together. But from a practical standpoint, it's not possible, because it would take so long to do it, and millions of dollars, and you'd have to have the right type of lab, and it would take so long to get that done. To give you an example, to produce an amino acid, one amino acid takes about 3 months of 24-hour-a-day chemical reactions in the laboratory. Okay, and so you know, a strand of DNA is multiple you know, amino acid and protein sequences, so you can see how long that would take. But while it's technically possible to do it, it's not, you know, it's not probable. And that is all I have on that. And once again, what I would like you to do is in the chapter concerning, in your book concerning DNA, there are questions 14 through 20, which is on page 209, short answer questions. I would like you to work on that. You should have enough time left in class to be able to complete that before the end of the period. And beyond that, I hope you guys have a great day.